So, welcome back. Today I am going to be going over DNA replication as a process. I'm going to go through it pretty quickly. We've covered this in class already, so this is just a backup lecture for those of you who'd like to review it or for those of you who missed class the first time. So the process of DNA replication is the process that takes one copy of our DNA genome and duplicates it to give us two exact copies. Being human beings, we have 23 types of chromosomes with two copies each because we're diploid. And so at the end of this process, we have two copies of all of that. So we get two full sets of genomes. Now we use this for cell division, and in mitosis, we break it off into two identical daughter cells, because each of those cells need all of the DNA, obviously. And in meiosis, we break it off into four gametes, with each of them having halvesies of a full set, so that we can make babies. So this particular process is occurring in the S phase of the cell cycle. Now, looking at how it happens on the largest level, we have here modeled a chromosome. So this is the entire length of a single chromosome. Instead of doing the whole thing in one fell swoop, there are areas of work being done simultaneously. So this process of replication is happening in multiple places along the chromosome at the same exact time. These areas of work are called replication bubbles. The benefit of doing it this way is that it's faster, right? So it takes much less time to do it in little pieces and then connect them than it would to do the whole thing in just one run. Okay? So these replication bubbles increase the speed at which DNA replication occurs. Within the individual bubble, the place where the actual replicating is happening at that moment is called a replication fork. It's like a fork in the road. So once you open up the DNA, it makes this fork shape, and that's a replication fork. It's a fork where replication is happening. Makes sense. Okay. So when we're talking about the workspace, we're in a replication bubble working at the replication fork. The overall goal of this process is to take one set of DNA and end up with two sets of the same DNA, identical copies. This process is done in a semi-conservative nature. What that means is half of the new DNA is created from the old strand, and the other half is created completely new and fresh. So the old pre-existing strand is called the template strand. It's quite literally the same exact strand that you started with. Right? We just ripped it off. Okay? The nascent strand is built through this process of DNA replication. So the nascent strand did not exist before. It's brand new, an exact copy. Okay? And so you have these two nascent strands in the middle of the process, and you have your two template strands on the outside. Now, due to the nature of DNA base pairing, right, these new strands are always replicated properly. Right? And so this will always be this. Right? They're inversed, but they're identical. So that's good. The enzymes involved in this process are listed here. Helicase is what's responsible for cracking open the DNA by breaking the hydrogen bonds between nitrogenous bases. So it's leading the charge here. Right? This is creating the replication fork. DNA polymerase is coming in to that now open space. It is reading the exposed DNA nucleotides, finding the complementary nucleotide, and then building the nascent strand. So DNA polymerase is what's building the nascent strand using the guideline of the template strand. Okay. Ligase is an enzyme that comes in on the backside of the process. Okay. And so there's going to be gaps to fill. One, the gaps between the different replication bubbles need to be filled. And as you'll see in the process ahead, there are spaces involved in this storyline that need to be filled in after the DNA polymerase runs its course. So the ligase comes in and finishes the job by filling up the empty spaces that need base pairs. The topoisomerase runs in front of the helicase, right? Because as the helicase is spreading open the DNA strand, okay, in front of that process, the DNA strand is now getting really tense, right? Because if you think of it pulling like strings, right, if you loosen the backside, it tightens the front side by pulling it, right? And so that's bad for the DNA. You don't want that tension in the DNA can break. So topoisomerase is up front, kind of just loosening up that bundle so the DNA doesn't break with that tension. And primase is an enzyme that comes in at the beginning of the story and installs an RNA primer, which is just a little chunk of RNA that's going to get stuck onto the DNA strand and allows the DNA polymerase to grab on so it can start rolling. So DNA polymerase cannot fit directly onto the DNA strand without this RNA primer to clink onto. Right? It's kind of like a zipper mechanism, where if you think about like the little chunk pot that the zipper goes into, and then you can zip, the RNA primer is like the chunk pot, right? And the polymerase is like the zipper. Right? General overview of steps. First thing that needs to be done is the DNA needs to be opened. Helicase does that. After that's open, the DNA polymerase comes in and creates the complementary nascent strand, builds the new DNA. Once the new DNA is built, you need to connect the unconnected, completed strands, fill in the gaps with ligase. And then I have proofreading the DNA at the end as step 4, but realistically it's happening along the way. So the entire time this proofreading is occurring. So you have to make sure that the DNA is being built properly, because you don't want DNA that's bad, right? That's wicked bad, and then everything will be ruined thereafter. So we have a lot of mechanisms that come in and evaluate whether or not this process has been done correctly. Right? Make sure that all the base pairs match up the way they're supposed to. And that's it. That would be DNA replication. It'd be done. These are the steps overviews. Looking a little bit more specifically at how those steps occur, we have to break it down into 2 separate mechanisms. There is the leading strand and the lagging strand. And because of the properties of DNA polymerase, right, the shape of DNA polymerase only allows it to move from the 3' end of the DNA towards the 5' end of the DNA. And so the helicase is moving in one direction. The strand that is 3' to 5' in the same direction as the helicase is moving is our leading strand. The strand that's going the opposite direction of the helicase is our lagging strand. And now the main players for both strands are exactly the same. It's just the way in which the nascent strand is built is slightly different depending on whether we're looking at the leading strand or the lagging strand. So the leading strand is really straightforward, right? The leading strand, this is going this way with the 3' to 5' going this way, right? So we're going in the same direction as the helicase. So the helicase has opened the DNA at the replication fork. Right? RNA primase has come along and plopped on our RNA primer right at the beginning of this leading strand. Right? DNA polymerase has come and fit on to the RNA primer, and it is ready to rock and roll down the length of the template strand, creating the new nascent strand in complementary base pairs. Now, this DNA polymerase can just literally follow the helicase all the way down because they're going in the same direction. So the helicase is opening it up, and the DNA polymerase is following along, building the nascent strand behind it. That's it. Nice and straightforward. Right? So the only little thing of interest here is that once this strand is completed, right, this RNA primer needs to be kicked out, and then that little gap needs to be filled in. Because the DNA polymerase couldn't read this little starting gap, the RNA primer was in the way. So you have these little base pair sections where they're not paired up yet. So once that RNA primer is bounced out, ligase comes along, reads those little naked base pairs, fills them in with the appropriate complementary base pairs, and then links it to the completed strand. And then you're done. That's it. Leading strand complete, right, in this replication bubble. Okay? So cool, done. The lagging strand working the same way, but in little tiny chunks. So in this model, the helicase is still going in this direction, right? Now I want to read only. Now we have to wait a second. There it's back. So the helicase is moving in this direction. Boop, boop. But our DNA polymerase has to go against that current. So instead of jumping right behind it and just rock and rolling the whole time, the DNA polymerase jumps on once a little bit is open and then builds a segment. A little bit more gets opened, DNA polymerase jumps on and builds a segment. A little bit more gets opened, DNA polymerase jumps on and builds a segment. Okay? So these little segments are called Okazaki fragments. And they're roughly 200 base pairs long. Okay? So every 200 base pairs that the helicase opens up, primase comes in, pops on a primer, and polymerase does its thing in this direction. Right? Boom, 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 boom. So we're building this strand in the opposite direction, little by little, as helicase is going along. So this is a little bit behind the leading strand, hence it being named the lagging strand. But it's a lot faster doing it this way than waiting for the helicase to open up the whole thing, putting an RNA primer at that other end, and then rock and rolling the whole length. Right? So this is the quickest way to do this, considering the limitations of DNA polymerase being able to move only from 3' to 5'. Again, Okazaki fragments, boom, bing, boom, boom. So ligase fills the gap, and now that one's done too. Cool. So at this point in time, both our leading strand and our lagging strand would have their complementary nascent strands built, and the only thing left to do would be fill the gaps between the different replication bubbles. Right? And so the ligase will fill those gaps too and make those connections. Boop, boop. And then you have your whole big new strand, whole chromosome copied. Done. Bing, bang, boom. You do that for 23 different chromosomes, you have yourself your whole genome copied over. So the last thing to talk about is the proofreading of this whole process. So again, this is happening the entire time that these things are being built. So there's a bunch of different proteins that exist that come along and effectively attach themselves to these DNA strands that are being built. And they just run down along the length of this new strand. And so if there are any base pairs that are incorrect, say there's an A paired with a G along the way, that shape is going to be incorrect. Right? It's either going to be a little bit too wide or it's going to be a little bit too narrow, and it ruins the smoothness of the flow of this DNA. So the protein is expecting a certain smoothness, and when it comes across one of these variations, whether it be too wide or too narrow or too janky, right, it identifies that as being a problem. Now, because the replication process is semi-conservative and we have that original pre-existing template strand, that protein knows which strand is correct and which strand needs to be replaced, right? Because otherwise the A or the G might have been the right one. However, because we know that the template strand was the one we started with, that would be the right one. So the G will get thrown away by G, and a T will be grabbed and plugged in and fixed. Right? And so by this mechanism, we can ensure that our DNA is copied fairly properly. Right? And so this process makes mistakes quite often, actually, because it's just speeding through, trying to get it done as fast as possible. But our proofreading mechanisms are really good, and so most of those mistakes are caught and eliminated. If really large mistakes occur during this process, the cell will just trigger apoptosis and it will be destroyed. Right? Occasionally, mistakes slip through. It happens. They fall through the cracks. And that's how we get things like point mutations. Right? And so without these kind of issues or these variations, without these mistakes, we wouldn't have any of the different alleles that we have in our gene sets, right? Because they're just little mistakes. And so, like, instead of having brown eyes, you end up with blue eyes. Or instead of having fingernails, you end up with no fingernails. That would be terrible. Can you imagine having no fingernails? How awful that would feel? Ugh. Ugh. That mutation better get caught by our proofreading mechanisms. But the other ones can stay. Okay? And that's it. DNA replication complete. Pretty straightforward, I think. Right? Yeah, definitely. So, I hope you had a great time listening to me talk about this. I had a great time talking about it. Have a fantastic day. Bye.